I'm Allison Johnjack, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
Welcome you to the July 21st virtual brown bag. Our July season is, of course, the last growing season updates brown bag before we will meet in person at the Summer Field Day and Trade Show on August 9th. So for a little bit of housekeeping, I have transcripts on. If you're not seeing those, click up in the settings and you should be able to see the live transcripts. Go ahead and keep yourself muted unless you are asking a question. I do like to encourage people to ask questions live and throughout. I think both verbal questions, or if you type questions in the chat box, you can either set that to everyone or you can set it to Allison Johnjack. If you set it to just me, then I'll ask that question anonymously on your behalf. We're going to have a couple of polls in today's session, and if you're on mobile, that might not be the easiest, so feel free to type your thought or share your thought verbally if your voting's not working. And yeah, I'll go ahead and hand it over to Tom for a quick welcome from the Growers Association.
Oh, well, thanks, Allison. We just welcome everybody to our July brown bag seminar. We do these routinely throughout the year, and we try to make the topics timely to what's going on in the growing season. Allison does a good job of that, working along with the education committee. So we are excited to continue to provide this service to growers. And actually, as I look at the participants list, it's pretty much across North America. So we'd welcome you to today's brown bag. Just one quick reminder and announcement is that, of course, the summer meeting, field day, and trade show is coming up August 9th. Allison mentioned that. We will be at the research station again this year. We expect to turn out vendors and growers. We would encourage you to get your reservations in or order your meal tickets so we can make sure we have a good count. We have some new things to show at the research station. We've got 12 different projects going on that will be highlighted during the mini clinic on the marsh programming throughout the day, and hopefully we'll have things of interest to anybody and everybody that wants to participate. And of course, it also gives growers the chance to have conversations with one another and really do a lot of networking, in which we encourage as well. So it's a major event for us, and your participation is important for us to have a successful day, and we hope to see you there. So with that, Allison, I'll turn it back over to you, and looking forward to hearing from today's speakers.
Yeah, fantastic. We are going to have today our usual observations from the field, but due to a lot of the interns being on vacation, several of our usual suspects in the scouting world and the IPM world were not able to join because they're actually out scouting. So I'm going to give just some quick observations from the field. Then we're going to have two what I think will be very useful and informative presentations. I have them in reverse order on this slide here, but Jed Cahoon is going to give us a lot of information on new plantings, best practices for making new plantings, fertilizing and weed treating new plantings, the whole gamut. And then Christelle Guedot is going to give us information on flea beetle updates. So that's going to be everything from life cycle questions to what chemistries are our best bets right now to nematode projects that are ongoing. So everything new plantings. And then also Jed was going to have a little bit on tank mix information as well. And then everything flea beetle. And then I will hand it over to Christelle.
Well, thanks everyone for coming. Thanks, Allison, for putting this together. And I will just start with a little follow-up on Jed's talk, and I'm putting that in the chat right now. But this is a link to a video So we do, as you know, we do a lot of fruit work, and Leslie, Amaya, and I have been putting together webinars over the years that started around COVID for the other fruit growers. And one of those webinars is all about tank mixing, and it's by Jason DeVoe, who's known as the spray guy. So I put the link here to that video. If you go onto the Wisconsin Fruit News and you look at some of those videos that we have on the YouTube channel, you're going to be able to find videos like this one that could relate to some of your questions. So there's another one from Jason DeVoe on sprayer calibration, if you were interested in that. But I think this one is really useful to think about tank mixing. So I just wanted to put that there. The other, while I'm sharing, comment I wanted to make is on diazinon. You have probably seen now the publication or the article, I mean, that I put together for the lack of supply for diazinon. Sure enough, when you put an article like that is when the companies reply to you or other people. So in this case, it was CI, that was Katie Gantus, that was able to get through to them after I emailed them, I don't know how many times, and called them. But Adama has said that they will be continuing production. So they have a supply issue, but there should be more production coming. So anyways, you're going to see another article that's going to talk about the most likely recontinuation of the supply, even though right now there was a discontinuation of supply on diazinon. And then I just wanted to follow up on what Allison mentioned about the late emergence of flea beetle or early emergence this year. I think for the most part, from what I saw from your responses and what we saw in the field, we're on par with what we have, and you'll see it in our data of that kind of July 9th being the week where we start seeing a couple of flea beetles. So I'm not too surprised that some people have not seen them yet. We have not seen them yet in our cage trials with the nematodes, nematode trial that we're doing. We have seen just a couple this week in the large field trials that we're doing with nematodes as well. So we're at the beginning as expected. And I'll show you some data from a little way back when we looked at that, when we were doing seasonal phenology. So now with all these quick updates, I just want to start into this presentation. So I was asked to give this presentation that was actually given by my postdoc at the time, Ben Jaffe. I forgot, in 2019, maybe somewhere around there. And it's work we did in the late 2010s on flea beetle biology. And I was asked at one of the board meetings this year to kind of give that talk again, because I guess somehow there's things like that we present, but not everybody was there. And so there's sometimes a need for a repetition for that. So you might have seen this presentation, but I'm going to tell you what we know now more about flea beetle biology. And so again, this is the work done by Ben Jaffe, who's down here. So when we started this work, and even now, so flea beetle in general, I'll say, is native, and it's not just in Wisconsin and in Cranberry. Here we call it Cranberry flea beetle, but it's actually called red-headed flea beetle, technically by the Entomological Society of America. And it's widespread in the eastern US. So I have colleagues that are working on flea beetle on the northern east part of the country. There are people south of here. So there's a lot of people that are looking at flea beetle, and really everybody is seeing as a resurgence of flea beetle in the last decade or so. And so there's more and more people interested in working on flea beetle and understanding better. And what's interesting too is that what we see in Wisconsin is not what they see down south. So here we are not exactly sure that what information they might put out, and none of it is really published. They're barely starting to publish now. But what we're finding in extension publication from nurseries and other commodities like that are dealing with flea beetle was not really something that was matching what we were seeing. So we weren't sure. We didn't know how many life stages there were. We didn't know how long they lasted. We still don't know about their fecundity. Where are the eggs laid? We don't know much about their seasonal phenology, etc. So those are a lot of the questions that we had going in that we couldn't pull from extension publications that we're finding from the nursery world. So this is trying to understand this life cycle. So we're trying to see what we have here, and then trying to get a little bit more detail on this in our region, because it's not going to be the same everywhere. So for example, here, what we suspected is that we have only one generation per year of flea beetle, and down south, they can have 2 or 3 generations. So what is that? What is the kind of differences that we see there? So we're not comparing with them, but what is it that is happening here? So what got us really interested is the egg stage, because we know the egg is the one that flea beetle are overwintering in. And so we were trying to figure out what are the requirements for this egg diapause. So what we have looked at here, so this is the temperature in Celsius, and this is the % egg hatch here. And so what we found here, if we keep those flea beetle, and we did that, we had some flea beetle we collected, we let them lay eggs, and some of them, of those eggs, we stayed at 20 Celsius, so that will be 74, 72 Fahrenheit, 72. And then some we dropped down to 10 Celsius, 5 Celsius, and 0.2 is the lowest we could get that's freezing in an incubator at the time. And so what you can see here is if we kept our flea beetle, which is pretty much what they can do down south, if we left our flea beetle, the eggs at room temperature, they would not hatch. We have very, very little hatch, egg hatch. But if we go down to 5 Celsius or below, we have optimal egg hatch. So we have this cold temperature requirement for those eggs for them to be able to then emerge. And they were left at those temperatures for 15 weeks, which then led us to what length could we go? And really what we found is that we need those 15 weeks, 15, 20 weeks, somewhere around there, but 15 weeks is ideal for getting at 5 Celsius, for getting optimal egg hatch. So this is the requirement that we have we don't know exactly what differences we're going to find between our populations and the populations down south. It could be that there's what we call ecotype, where they're going to be the same species, but really based on their environmental conditions and their location, they are much more different than we would expect otherwise. Or there could be that it's just because of the photoperiod that they're experiencing, the fact that they emerge in early July, they already passed the solstice. And so maybe the triggers from those photoperiods is already there to trigger that long overwintering diapause for the eggs. So something we're going to investigate in the future. So we also wanted to see how many larval stages do we have. We were suspecting maybe 3 from what we've seen, but there was no research that looked at that. So what we did here, and what Ben and the student Stephanie Rink did on this, is looking at the head capsule. So that's here you have the ages of the larvae, you have the numbers, the length of the larvae. The length is not always the best measure. The best measure is the head capsule, so the head really. And you can look at the width or the circumference of the head. And when insects are going from one instar to the next instar, this is the most conserved way of looking at stages at different instars, by looking at the head capsule because of their molting that happens. So that's what we looked at. And what we found is that between 1 to 7 days, we have one type of size of the head capsule. Then 14 to 21 days, we have a second type of head capsule size. And then after that, 28 to 35 days, yet a third one. So we have at least here 3 instars of the larvae. What happened is we had a lot of mortality after 42 days of having them in the lab. And what I suspect really is that we can control the temperature really well, but we have much harder time being very consistent with the moisture when we are in those incubators. Sometimes they dry up too fast, or we just put water in there and we're not able to really maintain a very constant humidity level. And this was also at the time where my postdoc Ben found a new job. So there was not somebody that was really in charge of keeping track of that. So we were not able to go all the way to the adults. And we had one pupa that we found at 51 days. So what we suspect is really that we have 3 larval instars, and then we have the pupae after that. So if we recap this, we have that we need about 15 weeks at 5 Celsius to have the eggs hatch, and they will hatch in about 3 weeks of having this after this temperature requirement. So you do 15 weeks, 5 Celsius, and then 3 weeks later, they'll hatch. Then we have 3 larval instars. I only represent 2 here, but there's about 3, there's 3 of them, and they last about 3 weeks total. And then we don't know exactly how long that pupal stage will last because we only recovered 1. So we're going to have to try to get back to having a colony in the future very soon here, hopefully. So that's what we did on the larvae and the eggs. And then we were interested in understanding more the feeding patterns of the adults. We know that they feed on the leaves, and you can see on this leaves up here that they feed, they kind of feed between the veins, and they don't go through the leaf. But what is it that, where are they feeding? Is that density dependent, how much they're going to feed on? And those kind of questions from the adult feeding. And do they feed on fruit was another question. So here, what we have is we looked at the percent decrease in berry weight after 7 days. So we put them entirely on fruit. And you can see here that we would have, oops, sorry, here's the mouse. So we have the control here where there was no flea beetle, and then here we put some flea beetle, and what we have is the percent decrease in berry weight. So you can see that they feed on the surface, mostly of the fruit, and this is supposed to be a video, but it doesn't work anymore on this new computer. But what we found is about 8.5 to 25% fruit damage. So the number of fruit that were with damage on them. And they tend to focus on one berry. Once they start on one, they stay on that one. So that's when we showed you that several years ago, that they are able to feed on the fruit. And we're seeing some of that in the field as well. And then we wanted to test different densities. And we also wanted to see where they were going to feed on the leaves and on the fruit. So what we did here is we look at the percent again of fruit that were damaged and the percent number of leaves that were damaged. And we put at the x-axis a different number of beetles in the cage. So we had either 0, that's our control flea beetle, 5 flea beetle, or 20 flea beetle in here. And this was going to be on the y-axis, the percent fruit that are damaged, and the y-axis on the other one, the percent of leaves that are damaged. And so, of course, you can see that there is an increase in the number of fruit or leaves that are damaged as the densities increase. So that was not too surprising, but sometimes you can reach easily a plateau with those kind of densities. And here we still had an increase. We have more densities in the next one. And this was done all in the field on this part. So here what we looked at is the percent leaf damage on the y-axis, and again, the number of beetles per cage. And we looked at where on the uprights they were feeding. So was it 0 to 2 centimeters from the tip, so the very tip of the uprights, a little below 2 to 4 centimeters from the tip, or much below that at 4 centimeters from the tip? And you can see that whether we have the low or the high density, and this could be density dependent, if you have too many of them on the top of the upright, they might move down a little bit more. But we see the same consistent pattern that they feed on the very tip of the uprights. What we think might be happening, and that's from talking with my colleagues south, is that it seems like they're just going up towards the light. When they tested it on ornamentals with new leaves and old leaves, and they just were going, there was no difference between new and old leaves. It was just a question that they're going towards the very tip of the uprights. They're attracted to the light. So maybe that's what we're seeing, and that's what we're kind of expecting here is happening as well. We did lab assays, and in this case, we did densities of 1, 5, or 10 flea beetle, and we put 3 to 4 uprights. So the trend is the same that we saw in the field. We still have an increase in the percent leaves that are damaged with the number of beetles that are put in there. And when they have no access to the fruit, they feed on more leaves, was one observation that Ben also did. So again, this time in the lab, when we do 1, 5, or 10, and we see this a similar trend, but you can see here that that you can see here that you will have, with one beetle, you will have more on the very tip. But then when you start getting here with the 5 or the 10, it changes where they end up feeding. So they'll feed more on the 0 to 4 centimeters when there's 10 of them, but there's still quite a few that go below because there's a lot of them on just 3 to 4 uprights here, compared to in the field where there was a lot more uprights available. And then here, on the 5 per cage, there's also more that go to the 0 to 2 centimeters and not so much difference between the 2 to 4 and the 4 centimeters. So it is going to be somewhat density dependent, and the availability of fruit may play a role there too. So there's been previous work now switching to where the beetles are located that Sean Steffen's lab had done. So what they had found is that, putting emergence cages, you have a lot of them that emerge from the bed, even though some emerge from the dike. So that's data you've seen from a long time ago. And so we wanted to see where in the beds they were going to be. So we looked at the eggs, the larvae, and then the adults with sweep sets and emergence cages. So on the eggs and the larval phenology, we wanted to see with soil cores, and we did the soil cores, they were 30 centimeters by 8 centimeters, so 30 centimeters deep and 8 centimeters diameter on the soil core. And we did this every other week, from May through September, and then again in November. And then we split that 30 centimeter core into 2 sections. One section was 0 to 15 centimeters, and the next section was the 16 to 30 centimeters to see if they were deeper in the soil core. But we couldn't do every centimeter. That was a tremendous amount of work. And then we did 1 meter from the dike or 10 meters from the dike to see if there was some kind of an edge effect. So here is the total number of flea beetle collected. So it is not an average per soil cores because we had a lot of zeros. So this is giving you the total numbers. So in black, you have the eggs, and in gray, you have the larvae. So we started in early June. I'm surprised of what this means down here. Sorry. So we started in early June, and we didn't see any in early June. We weren't able, it was May. Yeah, we started May 14th. That's what it is. And we didn't find any, but it is really hard to see the eggs. And at this stage, we suspect that we only had probably overwintering eggs. And so it doesn't mean that there's none here. It means that we didn't see any. And then June 19th is where we really start seeing a lot of eggs. And they could have been too that we gained a little bit more expertise with all the undergrads we had that were helping with this in finding the eggs in the soil. It's really hard. I'll show you a picture after that of seeing those eggs. But we found a lot of eggs on June 19th, and we started seeing some larvae. And then as you can see, we have fewer eggs, more eggs again in July 17th. And then after that, it's probably the eggs that are going to overwinter that we see from August 14th all the way until November 15th when we went back to check. So larvae, we only detected them June 19th until August 1st. So that's the window that we had for the larvae. And this is the eggs. So you can imagine when you have all those little grains of sand and everything, finding a larva, an egg, that small is really hard. And it took a lot of time to get ourselves comfortable with that. So looking at where the eggs are located, this is the distance from the edge that I mentioned, 1 meter or 10 meters, and the average number of eggs that were collected on the y-axis. there's no difference. We saw maybe fewer that were at the 10-meter distance from the edge of the bed, but really no significant difference. So they are laid at 1 meter or 10 meter. It doesn't seem to be any trend there. And then in the depth of where those eggs are laid, that was interesting because we found fewer eggs, significantly fewer, at the 0 to 15 centimeter depth than the 15 to 30 centimeter depth. I can't tell you how far down in the 15 to 30 centimeter depth, but that tells you that those females are burrowing all the way down to at least 15 centimeters to lay those eggs by the roots of those cranberry uprights. So that is very deep in the soil, which means that they're very well protected from any cold temperature or anything like that, and they're able to go through our winters easily. And here, if we look at potentially at 1 meter and at 10 meter, are there differences of how deep the eggs are laid? And there seems to be a little bit more digging that is happening potentially at the 10 meters. It's not necessarily something that will be very significant anyways, but interesting to see that potentially they are going to put those eggs deeper in the soil when they're further into the bed than when they are at the very edge of the bed. But there's a lot of, as we know, patchiness in those flea beetle, and so there could be patchiness in those eggs as well, obviously. Then we did the adult phenology. I'm going to check on the time. Yeah, I'm late. So I'll skip on the adult phenology. I'll just go quick. We did emergence cages, and this is what we got from emergence cages. We didn't see a lot of beetles emerge. Emergence cages are so small when you think about the patchiness. But when we saw them was all the way until harvest. Harvest is the bottom line. And we saw them that year. We saw them a little later around July 10th. When we did this with sweep netting, but we didn't do that in the bed. We did it either in the dike, on the dike, or in the wild. So in this graph here, you would have the dike in the light gray, and the wild habitat would be kind of like the set-aside areas that machinery set on. And we were sweeping there, and we still found flea beetle there in the weeds and stuff. And that would be the darker bars. But you can see here, this was 2018, that July 9th, just like you guys reported in the poll, is when we saw the first adults. So I think we're on par with what the data that we usually see is that first week of July, sometimes second week of July, or second week of July, sometimes third week of July is when we see our first adult emerge. So I'm not going to go over this, but this is what we know. But what I will say is that there's still a lot more that we don't know. And there's so much that we need to ask. And so with those colleagues that I have down south and more on the northeast, we are putting together right now a specialty crop research initiative grant that will allow us to ask questions about the basic biology, trying to understand. What I'm really interested in understanding is, are there, how many eggs are they laying per females? Are the females, once they mate, somewhat not able to mate anymore and kind of be refractory? Is there multiple matings? Is there just one mating per female? So all those are questions that are really interesting, that are also going to feed into really going back to understanding this sex or aggregation pheromone that the growers in nurseries are really interested in, to try to have better monitoring tool and potentially managing tools for sex or aggregation pheromone for, say, mass trapping or things like that. We're also going to be looking at plant volatiles to see if we can have a better tool for females that could be attracted to. I don't know, maybe Dave has a comment on that as far as what he's seeing with that for those as well.
Sure, I'll just chime in really quick. I mean, I guess the big thing just comes down to timing. You know, venom for a lot of growers in the state of Wisconsin is going to be one of the very few options available deep into August, because, like, just for example, from one handler's perspective, a 14-day PHI for some of that fruit compared to many others. And so even if they were venom and Actera were equal head to head, if you were worried about flea beetle, it would make more sense to lean on Actera, which is typically restricted more heavily For example, on the ocean spray side of things, a 45-day PHI, it would make more sense to lean on the product that is restricted more heavily from a pre-harvest standpoint and then save venom for when you get deeper into August. So that would be kind of my 2 cents on that whole situation.
And I really appreciate that, Dave, because that's something that is hard to keep track of. What are the restrictions that ocean spray will put on those products? So that is very helpful for people to know that.
I do have a few more questions. I'm gonna just, for the sake of those watching a video later that don't have access to the chat, Dave's research had shown Actera is making impact on flea beetle at that kind of moderate level. Alticore, we're going to see it as more minimal and would rate it as poor. Becky Heck adds that scorpion is labeled for cranberries as well, similar to venom, but it's a liquid formulation and so easier to handle. I have not personally read the label on that one yet, but I look forward to. We had a couple more questions, one from Cass asking more back on the life cycle and phenology side. Do we know what the plant response is when it's being attacked by flea beetle?
The plant response. Oh, you mean the long-term plant response?
Yeah.
No, we don't. I mean, that's something that I actually wanted to look into, but we haven't been able to do that. But that's something that growers are saying they see the impact that it has on the next year's growth. I haven't seen it, so I don't know what impact it would have in fruit production. That's something that I was really interested in looking at, and unfortunately we weren't able to do it this year. But yeah, we don't have that documented, but yeah, something that growers are experiencing.
Answer a little bit anecdotally that when the new growth, the current year's growth is damaged as they're seeking the tip of the plant, when you go out next year, you see reduced new growth, but that's only anecdotal. That's not documented at this point. Also had a question about can we talk about the nematodes a little bit? I know we've got trials at a couple of locations this year, and I know we're kind of doing more and more work on that front. So do you have an update on that front?
Yeah, so I mentioned that very quickly earlier on. So we did all the applications. So some of it is in collaboration with Dave Jones and Ocean Spray and of course, grower collaborators at 3 marshes where we're testing the capsinum, and then we're testing the native nematodes that Sean Steffen's lab is providing. And then we have a control. We have been doing now the 4 applications have been done in those beds, and we've been sweeping last week and this week. And so far, just a couple flea beetle have emerged at 2 of the marshes, and then 1 marsh had a lot of flea beetle. So that's interesting to think about, is that it varies where we're seeing small emergence and some other areas where there's a lot of emergence. So that's going. We'll see how the data holds up as far as like the whole season-long data. And then we also have a cage study where we're looking at 3 of those commercial nematode species, plus the including the capsinum, plus the native nematodes from Sean Steffen's lab. And as of now, in those cage studies, we don't have any beetles that have emerged, but we're also sweeping next to those cages, and we are not getting any in the sweeps yet either. So we're at a marsh where, even though it's a marsh with a lot of historical pressure, as of now, they have not yet emerged. So again, we're in the early emergence in some areas. For some reason, there might be, some kind of microclimate that plays into it, and some of them have emerged more. But for the most part, the emergence is at the very beginning still. So that's where we're at with the nematodes. But we're hoping we're going to have good data with that and something to provide to the growers next year at Cranberry School.
Fantastic, thank you. I think I'm seeing a trailing off to the questions that are coming in. So I will give people one more opportunity to ask questions. But while we do, I want to thank, in reverse order, Christelle for all the excellent flea beetle information. I want to thank Jed for the new planting information and the tank mix follow-ups. Remember Christelle's video on the tank mixing practices as well. And then thank Tom and the Growers Association for helping co-host these with the University of Wisconsin-Madison Division of Extension. And I also want to thank all of you for joining and all of you who asked questions today. I think the questions really make these great. We'll see you hopefully August 9th at the research station for the Summer Field Day and Trade Show.